I'm going to talk to you about, maybe I'll come over here. Oh, let's see. About what Orig3n is doing. So our strategy is around the fact that a lot of the reasons that we're all here at this conference is that many disease and injuries can be addressed by cell therapy. And so we've chosen two programs that I'll talk in a little more detail about to do internally. It's cartilage and heart repair as our first two internal programs. We've also leveraging a one-of-a-kind cell bank to manufacture our cells for allogeneic therapy, to have cells ready off the shelf. And we've licensed induced pluripotent stem cell technology from IPS Academia Japan. And so our whole cell therapy program is built around IPS, starting with iPS cells. And the idea with allogeneic therapy, of course, is that by generating these special lines, we can basically pursue a number of programs simultaneously with increasing the optionality and decreasing preclinical R&D costs. So why iPS cells? It's an unlimited source of stable cells for manufacturing. They have the capacity to be grown both in 2D cultures as well as 3D cultures. And of course, 3D cultures are very useful in terms for manufacturing, and they can differentiate into any cell in the body. And then readily made from adult blood cells via reprogramming. So this has allowed us to build our bank. And of course, it's not controversial. It's a non-controversial alternative to embryonic stem cells. So it's really important to get the right cells. So we've actually spent a fair amount of time in our laboratory working on various differentiation protocols and really looking at different aspects of the cells that we're creating. And as a result, we have a proprietary patent-protected GMP compliant method for differentiating our cells. One aspect, so we've looked at two aspects. One is around the secreted proteins. So there's a lot of proteins that are secreted. In fact, there's more than 10% of the protein coding genes are predicted to be secreted from the cell. And I've listed some of the properties of those proteins that are secreted. There's growth factors, cytoskeletal proteins, proteins involved in wound repair, involved in anti-inflammatory, extracellular matrix proteins, as well as proteins that are antioxidants. So this group of proteins, of course, varies, is going to vary depending upon the actual cell type. But it's known that these secreted proteins actually play a crucial role in many physiological and pathological processes. So we've looked at the secreted proteins that the cells make, and we've also looked at gene expression patterns to look at them as well. And then also we've done animal studies where we take different types of cells and we've looked at efficacy in different models. So one of the problems with cell therapy is the fact that you have to have matched cells or your body is going to reject them. So this is why we've went with allogeneic cells and basically produced a large bank of cells that I'll talk to you about. And because, of course, the closer the match with immune markers you have, the less chance of rejection. So our cell bank, we collected many tens of thousands of samples, and we looked in our donors by profiling them for their HLA haplotypes to identify what are called the super donors. And these are basically homozygous, people who are homozygous for the HLA markers. And so as you see on the right, this one donor that's orange-orange, figuratively speaking, can match the person that's orange-green or match the person that's orange-purple. And so as long as they have one match, then it's known from both bone marrow transplant and organ transplants that this won't cause rejection. So we built this bank basically in a crowdsourced fashion. We went to public events like marathons and triathlons, music festivals, Comic-Cons. We'd set up a booth with phlebotomists, and we'd ask people if they'd like to donate some blood for our work. And so the entire bank is fully consented for all uses. So we're able to use the cells for our own research, for commercial purposes, and share them with others. And also in terms of the size of the bank and the HLA matching we've done, we know we have matched cells for 90% of the US population, as well as significant percentages of other global HLA haplotypes. So just advantages of our cells is allogeneic, of course, is going to be more efficient than autologous, and iPS cells and differentiated cells from iPS cells will provide an unlimited number of cells that you can control and purify and have frozen off the shelf so they're ready for multiple dosing, so it's more cost effective. And then, as I mentioned, we've optimized our cells based on their protein secretion and their gene expression profile to really match the application. So as I mentioned, we have two programs. One program is around cartilage repair. I think everyone knows that this is definitely a high need area. A lot of people suffer. A lot of people become immobile as a result of problems with their joints. And really, the other thing is the conventional therapies don't address the underlying loss of the cartilage. So they're all associated with helping people cope and give some, at least maybe temporary relief, such as physical therapy, anti-inflammatories, et cetera, that I have listed there. And then as a last resort, people go to knee replacement surgery as a last option. So off-the-shelf HLA matched cellular products are going to provide not only pain reduction and cartilage replacement that might stop the degenerative nature of the disease and provide actual repair of the cartilage. So I think long-term relief is possible. So we've done a couple of animal studies. We've initially done the medial meniscal tear model, which is a very popular model done in rats where you produce a surgical lesion. It's very discrete, and you can look both at inflammation as well as cartilage repair. And so we found with our cells, and I'll show you some of the data, that the cartilage damage was decreased with our cells. We looked at some cytokines with lavage of the joint and found IL-6, for example, was decreased. And it's a measure of both acute and chronic inflammation. And also that synovitis, which is a inflammation of the lining of the joint, was decreased relative to the vehicle. And here's some of the data. So these are panels of individual animals. So the far left two panels is an uninjured rat joint. You've got the femur and the tibia. And if you look at the dark blue, which is the staining of the cartilage, you can see in those two animals in the far left, they have a nice continuous cartilage lining the joints. And the middle four panels are the vehicle treatment. So these are 4 individual animals that had gotten the surgical treatment. And you can see the interruption of the dark blue. See, I have a pointer, maybe? Yeah. So you can see an interruption here and here and here and here of the cartilage. And then these 4 panels, these are 4 of the animals that were treated with our cells. And now you can see the cartilage is filled in nicely where the damage had been. You can quantitate the joint damage with a lot of different types of measurements. Here's 2, a total joint score, where you're measuring the amount of cartilage, and tibial degeneration, where you're looking specifically at the cartilage around the tibia. And so in both cases with our cells, you see a decrease of the damage, the degeneration. And then here's for inflammation. So IL-6 I'd mentioned, in which we see a decrease in IL-6, and the synovitis, which is the inflammation around the joint. We're seeing a decrease with our cells as well. And hyaluronic acid, which is one of the therapeutic options that people have now. This is actually a fairly low dose in these animals, but you see that you get increase or no effect in terms of inflammation. And in fact, if you give higher doses of hyaluronic acid, you start to get greater and greater inflammatory effects. So in terms of the cartilage program, we've developed proprietary methods for differentiation and handling our cells for cartilage repair. We found that they not only seem to repair the damage after this surgical tear model in the knee joints of rats, but also reduce the inflammation by measuring IL-6 and synovitis. So both anti-inflammation and reduced repair are going to be important properties for the beneficial treatment of osteoarthritis. Where we are, our status is we are doing some additional preclinical studies as well as preclinical toxicity studies this year, and plan on filing an IND at the end of 2020. So for cardiology, this is a program that we are collaborating with Harvard and the Harvard Stem Cell Institute. Dr. Rich Lee, who's there, both in his lab and our lab, we're developing suitable methods for cardiomyocytes and a very specific indication for cardiac disease. So currently, the status of cardiomyocytes generally is that they're not really electromechanically mature enough to be good, to have the right properties for cardiology repair. And so really the challenge there is to work on maturing these cardiomyocytes, especially electrically, to a greater extent so that there won't be as much of a mismatch when they're applied to the heart. So this, again, actually is probably about a year behind. We're going to start animal studies this year. We've been making actually a lot of progress in this area, and we're going to be filing an IND in 2021. So what makes Orig3n unique, just in summary, we're custodians of the world's largest cell bank for regenerative medicine today. We're using it for our own purposes, as well as happy to work with others who in other therapies. We're developing these homozygous HLA superdonor lines to be used as a source for cell therapies. It's uniformly consented and fully waived bank. We actually not only have healthy cells, but we also have disease. So we have a number of disease cells as well, which are very useful for modeling different diseases. As I said, we have two internal programs, one around cartilage repair, the other around cardiology. And we're open to, certainly open to partnerships if you want to contact us. And we believe together as a community, people who represented at this meeting, we can find treatments and cures faster using cell therapy. And I thank you for your attention.